outline of what I planning to cover today. So first, I will give you motivation for the use of Bayesian methods in computational biology, broadly speaking. And then I will cover two Bayesian models to study fitness of cancer subpopulations. So the first one is a Wright-Fischer model for time series. And the second one is a model for single-cell phylogenetic inference from copy number profiles. And if I have time at the end, I might talk a bit about a quick overview of the underlying computational methodologies. And just to make sure you can see that my slides changed.
Yeah, they're good.
Perfect. So let's talk about this, this motivation first. Why Bayesian methods in computational biology? To explain one of the main motivation is, I will first talk about the notion of calibration, more precisely, calibration of measures of uncertainty. So it's quite easy to, when you make a prediction, to just put some measure of uncertainty on it. But this measure of uncertainty could be misleading. So I guess a classical example is if you see that in a weather forecast, there was a claim that they were 50% sure that there would be rain tomorrow. The property that we would like about this kind of forecast is that 50% of the time they were right. So that's kind of what you can see here. It's a classical example that if you look at, for example, the National Weather Service, they are quite calibrated. So when they say they're 50% sure, they're right about 50% of the time. Whereas some other sources like your local TV meteorologist, for different reasons that you can imagine, they're sort of a bit misleading in their prediction that when they say they're 100% sure that it will rain, it actually only rain about 70% of the time the day after. So in statistics, that's kind of the main thing that we do. One of the main things is to try to get calibrated measures of uncertainty. So classical methods that you learn in the introductory statistical course will often assume that we're estimating a vector. So a vector is the thing that we're trying to reconstruct, and we try to get some uncertainty quantification on this. So when we have a vector, we're in good shape because we can get the right substrate to do a normal approximation, and then that's how a lot of classical measure of uncertainty are based on that. But in computational biology, we often need to estimate things that go beyond vectors. So we encounter things like networks or phylogenetic trees or conformations, and they are not a good fit for the classical ways of estimating uncertainty. And that's one area where really the Bayesian framework shines, is that it can give us calibrated uncertainty beyond vectors. So some uncertainty over trees, for example. And one of the key nice property of then this notion of uncertainty that come out of Bayesian model is that they can be guaranteed to be calibrated if the model is well specified. So if you put enough care in building your model that it's a close representation of reality, then you can have nice guarantees, even if you don't have very large sample size. So they're very nice guarantees. Another sort of catch, if you will, is that they might be harder to compute. So computationally, they might be very difficult. But often they might be the only viable option you have for calibrated notion of uncertainty if you're going beyond vectors. And so that explains a bit of the research foci that I focus on right now in my lab. So on one hand, trying to actually do this calculation, so having scalable Bayesian inference beyond vectors, and also to build tools to build approximately well-specified models beyond vectors. And I'll just give a minute or two to give you a broad overview, and then I'll focus more precisely on the topic for today. But just to give you an idea of the different kind of work that we do in my group. So one of them is to develop inference method that can deal with these combinatorial objects like trees and network, and actually do the calculation in order to get uncertainty estimates. And so if I have time, I'll talk a little bit about this at the end. So it's a new type of method that is based on non-reversible dynamics. And maybe for now, I will just leave it like this. If I have time, I'll talk more about this. But this is really what's enabling these study that I will talk about in the rest of the talk. So some recent uses of our non-reversible methodologies that allow us to do Bayesian inference at scale. So one of them is this single self-analytic inference problem that I will talk about today. Another application that came up recently is they used our methods to produce this new high definition image of the M87 black hole that came out last year. It's also used for plasma reconstruction. So these new computational methods are really pushing the envelope in terms of what we can dream up in terms of Bayesian models for large scale problems and large scale models. And then the other thing that we do in my lab is to develop tools and models that are good approximations of complex systems. So today I will talk about diffusion. So we're going to see a certain stochastic process that we can use to analyze the fitness of cancer. Another closely related project that I'm not going to talk about today, but you can look at this preprint if you're interested in, is another stochastic process that's used for to study population genetics, is jump processes. And I have a student, a former postdoc that are working on these jump processes. Today we're going to talk more about diffusion. So this project that the joint first author are Sohrab Salih and Faria Kabir, and here's the link for the paper. And some other example of complex model we build. So we build model for phylogenetic trees. So this will be the second part of this talk. So this is the joint first author are Sohrab Salih and Fatih Midori. And this is facilitated also with a new type of programming language called a probabilistic programming language that has been developed by many groups in the past 10, 20 years. And what these tools, they enable the modeler to construct this complicated probability model in a user efficient way. So instead of having to spend a whole PhD thesis on writing these implement in computer, these complex models, we can now write these models fairly quickly and prototype them. And that's very transformative for the field of Bayesian statistics. A lot of the existing probabilistic programming language don't support combinatorial structure. So one thing that we did is to develop a new probabilistic programming language that support things like trees and network. And that's also the tool that's powering this, fueling this second part of the talk on phylogenetic inference. So I'm not going to have time to go over this in detail, but you can go at this address if you want to hear more about this open source probabilistic programming language and use it for Bayesian inference in your favorite combinatorial space. So this is just a bit of a high-level introduction. Now I want to talk a little bit more specifically about this model. So to study fitness of time series. And the context here is something that lecture series that you'll be quite familiar. So it's intratumor heterogeneity. So as we know, as cancer cells proliferate, they accumulate more mutation than normal cells due, for example, to the activation of DNA repair mechanisms. And this creates several subpopulations with different fitness characteristics. For example, resistance to treatment or proliferation potentials. And so the motivation for this model I will talk about in the first part of the presentation is to understand this evolutionary process within a tumor. And part of this presentation will be to try to define a little bit more precisely what I mean by an evolutionary process, mathematically speaking. The experimental setup that motivates also the observation model for our work is patient-derived serial xenograph. And so here we start with a patient tumor that is engrafted in an immunosuppressed mouse. Then it is let to grow for several weeks. And then the mouse is sacrificed and the tumor is sequenced. And then part of the tissue coming out from this first mouse is engrafted in the second mouse. And this creates a time series where the observation are these, the statistic from these sequencing between passages. So to give you a bit more detail on the experimental setup, what is going on is more than one time series. So there are replicated time series that are used for example, for validation. And we can also vary some covariates, for example, drug intervention. So the color here, you see that the blue here are points in the time series that are subject to cisplatin treatment. And and then we can study how the fitness change from the part that are in the treated part and the untreated part. I will come back to that a bit later once we build a model. And then we can also do other things like taking two different time series and join them back. So mixing the biological tissue and to do prediction of what will happen when we do this, again, as a validation. So really, this is not just a line, it's really a DAG of directed acyclic graph of where these engraftment have been done, where each node is a mouse in that picture and the edge is an engraftment. Any questions on the setup? Or hopefully that should be clear. So what we're going to assume in the first part of this talk is that we have inferred a phylogenetic tree. And more precisely, we have taken the union of all the cells that have been measured using single cell sequencing, and that we build a tree, a phylogenetic tree on this on these cells. And I will talk more about the tree inference method in the second part of the talk. Just imagine for now that there is a structure like this, where there's a node that's the root, and then this shows the branching structure of so it's a cell tree. Now, another assumption that we're going to make that we can relax later is that someone, for us, partitioned the tree into subpopulations. So that essentially there were cuts that were done in the tree that subdivides the cells in the tree into different colors, different subpopulations. So I'm going to use colors for a subpopulation. All right. So this, for now, we assume that this has been done. Now, what is the model trying to achieve from this input data? So first, let's talk about how this information is translated into time series. So what we're going to do now, recall that this tree has been computed from the union of all the cells. But of course, experimentally, we keep track the origin of each of those cells from single cell sequencing. And so we can now go back and say, okay, for the first, the first engraftment, for the first mouse, how many cells can we find in population I, let's say. So population I is this region here. And so we see that there's a certain number of cells, 744, that are from this first mouse. And then we can ask this question for each of these other mice. And that gives us this sort of data, so integer counts for each engraftment event. So now the first objective for our model will be to reconstruct the population trajectories. So essentially what happened between these observation events. So you can see that I'm going from essentially a population to a sample. So so maybe essentially this at the bottom here are samples from this underlying population that I'm trying to reconstruct. So in this axis here, most of these diagram will be normalized. So this is a clonal fraction. So I'm going from absolute count to a series of numbers that sum to 1. So a point in the simplex at each point in time. And so again, so once we've, let's say, have some method that can reconstruct these, to interpolate, if you will, these trajectories that live on the simplex, our second objective, which is really the key thing that we're going at, is to interpret these trajectories and explain them in terms of simpler mechanisms. As usual, there are at least 2 competing explanations for these variations. So one of them is that there's genetic drift because these are a finite population, but most importantly, there is a bottleneck effect where at engraftment time, there's a small number of cells. So there's their stochasticity just due from, even if they were all of the same fitness, there would be fluctuation in their frequencies. And then there's a second explanation, which we're more interested in, which is the fitness itself, that some of these subpopulations just have more growth potential. So we want to build a model that disentangle and give us concrete quantitative estimate for these fitnesses. So how do we do this? At a high level, the model is organized like this. So it's just a hidden Markov model or a state space model that has the following directed graphical model. So we have the variable zi's. They're going to be at observation time to start with, and they take value on the k simplex, where k is the number of population or colors, and they are a list of k positive number that sum to 1. These are unknown. That's what we're trying to reconstruct. And what is known is these random variable yi, which are a list of k integers, the number of cells sequence of each population at that time point. And moreover, there will be a set of global or so-called static parameter, which determine the probabilities for these transitions. So really the inference goal, these fitness, they will live in that global parameter. So to give you a bit more details, we're going to assume a simple initial distribution, just a Dirichlet distribution with all parameter 1, so uniform on the simplex. The emission at the moment are simple, so they are multinomial based on ZT, so that can and should be relaxed in future version of the model. The more interesting bit is how the random variable ZT, its distribution conditionally on the previous ZT and the parameter. And so this is given by a Wright-Fischer diffusion. And that is the next thing I will review briefly. One thing to take into account is that this conditional distribution will depend on the amount of time that elapses between these 2 passages. In future version of the model, we will probably expand this time to take into account the bottleneck effect. But for now, it's just some kind of diffusion time, which is days rescaled by the effective population size. So let me give you a bit of, I want to explain what is this Wright-Fischer conditional distribution. So let's talk a little bit about, to start with, the Wright-Fischer model. And as a second step, I will describe the Wright-Fischer diffusion. So this first model is a model that is more mechanistic, but is a bit more computationally demanding. So we're going to approximate it in one moment. But for now, to understand the underlying simplification that we make at the level of population genetics, that's useful to talk about it first. So we're going to talk about a finite population, which are these circles, and we're going to talk about discrete generations. And I'm showing just two successive generations here, t and t plus 1. And it's a probability model. So I just need to explain how I can simulate generation t plus 1 given generation t. So I'm going to start with the first individual at generation t plus 1. And I'm going to simply pick its parent uniform from individuals in the previous generation. And I'm going to copy the color that I obtained this way. And as I do this for each independently, conditionally on the previous population, I get the next generation and I can iterate in this fashion. So that's the classic Wright-Fisher model. Now, let's talk about its modification that takes into account selection. So the Wright-Fisher model with selection, it is a similar story that we first assume we have the previous generation that has been simulated, and I need to simulate the next one. The key difference is that instead of picking the parent of each individual uniformly at random, I'm going to pick it using weights, a weighted sampling. And so the individual of color K, so K and the index a color or subpopulation, is weighted according to 1 plus SK. And so imagine that now these circles, they don't have all the same size. Some of them have smaller weights than another one. And then I would normalize all these weights and sample according to these weighted probabilities instead. And then as before, I'm just going to copy the color and repeat in an IID fashion. And so as you can see, this SK encodes the fitness of population K. So that's the key parameter that we're going to try to reconstruct using Bayesian tools. Now, the exact number of generation is unknown, and so and would be only weakly identifiable with other parameters such that the effective population size. So both because of identifiability concern and computational concerns, a common step is to then approximate the Wright-Fisher with a diffusion. So let me explain what is the Wright-Fisher diffusion. So let's make this thought experiment where we're going to let the effective population n sub e go to infinity, and at the same time setting the time between generation to 1 over NE. And at the same time, we're also going to normalize by n sub e to go from counts to frequencies. So this is some kind of sophisticated way of squinting that basically, of course, we know that these populations don't grow and don't have all these simultaneous dynamics, but there's sort of a leap of faith that if these counts are big enough, then the limiting object of this process is a reasonable approximation to the process I described in the last slide. So there's a similar kind of mental leap of faith that we need to do for central limit theorem. It's just a bit more complicated because we have to let these different parameters grow at a certain rate. But empirically, for some but not all cases, it's been found to be a good approximation of the finite Wright-Fisher model. And so what does it converge to? So it converges to a certain object that's smooth and nicer. And to explain that limiting object, I need to introduce the notion of diffusion or stochastic differential equation. So this often is presented in sort of heavy mathematical machinery, but the basic idea of diffusion is actually fairly simple. I can just explain it in one slide. So it's a way to explain, to describe generation of these curves that you see on the left here, where I say that if I want to approximate a generation t plus a small time interval, if I want to approximate that random variable, I use a normal random variable that is centered at the previous ZT. So ZT, think about it as like the number that encode these frequencies, plus a small change in mean that's determined by a function, let's call it mu, that may depend on where I am currently, what are the current proportion. And as a variance, or a standard deviation that's based on that function sigma of zt. And here for this delta W is just a normal, an IID normal. So this recipe here is essentially giving you a way, if delta t is very small, to generate these pictures of continuous trajectories that interpolate between frequencies. So that's sort of diffusions in general. Now let's specialize this to the Wright-Fischer context, and where mu and sigma take a specific form. I'll just say briefly that the reason diffusion are so widely used is, again, a central limit theorem argument. And here we applied this tool to the Wright-Fischer, and we get specific values for the mean mu, which are is giving you how much drift there are for each of the different k proportion. So it's an equation that may at first look a bit weird, but it's easy to compute. It's based on the effective sample size. It's based on the frequency of the population and times its coefficient, fitness coefficient minus the dot product of the two. And where that comes from really is just from normal approximation of multinomial. So there's nothing really deep here. And sigma is given by these value that involve this indicator function that's either 1 if I equal j and 0 otherwise. The key thing is that these, we have closed form for them, and they depend on these parameters of interest. So let's just step back a little bit and go back to our high-level picture of the model. So we have now this parameter theta, and more precisely is this list of k values s, and we're going to put a prior distribution on them, since we're using a Bayesian approach, and we're going to start with, let's use a simple prior, which is uniform on a fixed interval. That's much bigger than the values that the posterior value that we've observed in practice. So it's a sort of a weakly and uninformative prior that we're putting on these SKs. Now we're going to have a Bayesian approach. So we're going to get not just a point estimate on these SK, but a posterior distribution on these random variables. And so to give you some example of visualization of what these SK look like. So one of them is to look at the marginal distribution of each of those. And so for these different population, we get different values for these. We can see sometimes that with high confidence, that population in red, the E, is more fit than the baseline, which is clone C in this example. One thing that I will point out is that sometimes these pictures here might lead you to think that there's more uncertainty than there actually is. Just imagine if all of these posterior distributions shift jointly because of correlation, then you might actually be fairly certain that pairs are differently fit than this picture look like. So a more detailed representation of the posterior distribution is to look at each of these pair of distribution and ask what is the probability, the posterior probability that one of the coefficient is bigger than the other one. And you can see, for example, that for many of those, we have values that are we're fairly confident that there's an ordering, even though there seemed to be an overlap on that picture. So just a cautionary note on visualizing high-dimensional posterior distribution. It might be better to look at pairwise. So this is conceptually what we get out of this. Let me give you a bit more detail on how computationally we actually get these posterior distribution, because that matters. Often a lot of the challenge in modern Bayesian analysis is in the computational part. So let me talk a little bit about this. So at a high level, what we're going to do, we're going to alternate between sampling the fitness parameter, which are these global parameter in theta, more precisely SK, we're going to do that conditionally on the zk. So this step is fairly straightforward, so I'm not going to spend too much time on it. The difficult part is to sample the population trajectories conditionally on the fitness parameters. So that's a more high-dimensional problem. That's why it's more challenging. The basic tool that we used is particle MCMC, particle Markov chain Monte Carlo. It's a method to embed a particle filter within an MCMC algorithm. If you haven't seen sequential Monte Carlo method before, so it's a method that's specialized for time series. It's a good fit for this. Sort of accidentally, it turns out that at the mathematical level, it's actually very similar to the right Fischer model with fitness, except that the fitness changed over time. That's sort of just an accident. It's kind of a nice thing that recently there's been an interplay between how we do this calculation and the population genetics world, where some of these mathematical objects are actually similar. So in some sense, we're using population genetics idea to think about population genetics. But anyways, that's all a bit meta. What matters here is that we use particle MCMC to do this calculation. The challenging bit is that between these two random variables, because of the structure of the right Fischer, which we could compute these or approximate these posterior distribution if we didn't have fitness or if we had a very small number of population. But because neither of these are true, we need to do what's called a Euler-Maruyama approximation, where we introduce new random variable that discretize the interval between successive observations, and they use the same equation I showed you when I introduced the notion of a diffusion. So each of those are normal random variable conditionally on the previous one with mean and variance that change based on the previous observation. So because of these varying mean and variance, they're not jointly multivariate normal. So we need to use a particle filter to do these approximations. Now, one thing is that we cannot use tools from the particle filter literature as is, because particle filter, they scale very well if the dimensionality of each of these ZT is small. So if it's one-dimensional, two-dimensional, that works really well. If you get to the 10, 20 dimension, like some of our cases, this really gets to the case where vanilla SMC, sequential Monte Carlo, start to degrade in performance. And so we used a new solution for this kind of problem based on old block sampling ideas applied to this new problem. And the idea at a high level is to look at the current configuration of these imputed trajectory. We're going to pick at random a subset of population, let's say these three that I point with an arrow. We're going to erase the trajectory of their trajectory relative to each other, but not relative to the rest. We're just going to basically erase this. And we're going to resample only the part in white here. And so because we're dealing with a number of particles that a number of population we can control, so we can pick a small number, we can get a better asymptotic guarantees, or at least empirical properties compared to the full particle filter based on all the population. So that concludes what I wanted to say about the computation. Now let's talk a little bit about some of the validation we did for this method, this model. So a first idea that I briefly mentioned when I was talking about the experimental setup is that we can create what you could call de novo mixtures, where we take biological tissues and mix them and engraft this mixture into another mouse. And then since we've done inference for each of the 2 time series, we get estimates of fitness coefficients, and then we can make prediction on how this mixture will evolve over time. So what I'm showing here is, well, two things superimposed in black. These are what's called a forward simulation. So based on these value sk that I've inferred from previous time series, what is my prediction of how these dynamics will unfold? And in red, that shows the mean of these trajectories, clouds of trajectories, and then in blue, the observed point. So as you can see, the model does not claim to be able to do perfect prediction. So there is some width to this cloud of particles, and the main guarantees that it gives us is that it believes that the true trajectory is within that cloud. And as you can see, these seems to be calibrated based on these biological experiments. The other thing that we can do now, in terms of investigation of what happens when, for example, we change covariates. So in the future, it would be interesting to introduce covariates as basically some kind of GLM that control the generalized linear model that controls this fitness coefficient. For now, we did something very simple, which is to run the model twice, one for a time series that has, say, cisplatin drug intervention, and a separate run of the model where there's no drug intervention. And we can look at difference in the two posterior distribution over these SKs. Okay, so what we can see is, and we also have replicates. So we have biological replicates for treated and non-treated. So first we can see that here different rows would be different replicates. So if you look at only the untreated, there's the behavior is similar for the two replicates, and similarly for the treated one. And interestingly, there are big difference between the treated and untreated, as you would expect, but we can now quantify them. So we can look, for example, at these posterior summaries and look at just point estimate. Let's say we could look at more sophisticated stuff in the future, but let's just look at point estimate and order them in terms of the more fit to the least fit, and basically look at how this changed when we change the experimental condition to add the cisplatin intervention. And as you can see, there's a dramatic change, which you can sort of informally talk about as almost an inversion, but a big change in rankings. So I just want to talk briefly about some potential methodological direction for the future. So as I mentioned at the beginning, in order to get good Bayesian models, we need often to have models that are a faithful representation of reality. And this model that I've described has many limitations. But the nice thing about using this diffusion framework is that it provides a good framework to incorporate improvements to the model. So I'll discuss some of these potential improvements now. One of them, I think, is this notion of subpopulation. So right now, I mentioned at the beginning, we have to sort of cut this tree at different locations to get these subgrouping. And this is now at the moment done with a pre-processing step. So a deterministic function that has some criteria that we want to have from these cuts. And this is good enough for small throughput, or throughput meaning the number of subpopulation. But if we want a large number of subpopulation, an interesting direction would be to do joint inference over these cuts to basically add this uncertainty of what is a population. We know at a high level that we would like these population to be groups with different fitness coefficients. So there's sort of a well-defined notion of our desiderata, but it would be nice to incorporate into the Bayesian inference. There are also models that instead of having this artificial step of breaking the tree into chunks, look at the shape of the tree as a whole for inferring fitness. And so in the future, it might be interesting to apply these models, perhaps once we have a bit more data, more cells per mouse. Some other improvements, so richer evolutionary models, so incorporating mutation, potentially ecological process. Right now, it's only competition, but you could imagine having some cooperation maybe. An important one is to model these bottlenecks event, I believe would be a good addition to the model. And hierarchical modeling, meaning that a lot of these different time series, they're inferred separately, but there's an obvious ground here for hierarchical modeling. So lots of good projects for students that are looking for ways to improve modeling of these fitness time series. There's also computational aspects. So currently we use this very nice specialized block particle Gibbs sampler, but there might be ways to speed up the inference based on, for example, multi-level methods, which are quite popular in the stochastic PD literature. So maybe I'll ask here if there are some questions. And there will be more time at the end, but this is, maybe I have probably another 10 minutes or something like that. Maybe I'll try to talk a little bit about the beginning of this story, which is how to get the tree. So this paper is, in a sense, doing the first part of this pipeline. So at a high level, the input here is a matrix of copy number profiles for in the order of thousands of cells. And the output is a posterior distribution over phylogenetic trees. So the difficulty with using as input copy numbers is that it's a complex evolutionary phenomenon. There are many things that can happen. So we can have overlapping events that break typical independence, site independence assumption that we find in phylogenetics. So here's an example of a gain followed by a gain, and you can have overlapping effect as well, for example, overlapping losses. And so as a whole, this creates a complex combinatorial dependencies across sites. In the Bayesian literature, these are often called doubly intractable inference problem, in the sense that even if I instantiate the unknown random variable, I might still have it might still be challenging to compute the distribution of this one configuration, let alone explore a set of possible configurations. So one question was that, can we simplify the problem but still approach it with a full Bayesian inference, so i.e. with MCMC style methods? And the method we use for that is to use a certain transformation that relax the dependencies. So it's based on a certain notion of copy number change point. A key thing here is that it's going to be lossy. We're going to lose some information. And so the transformation is defined as follows. So we're going to, based on the copy number n, right on the left and on the right of each boundary point, we're going to look at whether there's a change in copy number simply. So here's an example here. So the output of this transformation will be this binary thing that here there was no change. So it's 0, and there was a change here, so it's a 1. So we do this for the across all adjacent bins, and we get a set of loci from this. Now, property of this transformation is that most of the scenarios that you can enumerate will satisfy the perfect phylogeny assumption once I encode it with this transformation, but not all of them. So it's not perfect, but it does reduce the dependencies quite a bit. Now, the key benefit is that we get efficient algorithm out of this. So at a high level, the one way to view what we're doing here is a bit like doing approximate Bayesian computation, but without the C, without the computation. So what do I mean by this? So what we want here is to compute the probability of the tree given the data. But in a sense, that's too hard because of the combinatorial constraints. So the idea here is to target instead probability of 3 given a deterministic function of the data that we can compute. And this is the first step that is done in ABC. So these statistics that are not sufficient. Now, the key thing though is that we're going to pick F carefully, well, the one I showed you here, so that we can use specialized computational methods that are based on Markov chains. In contrast, full ABC method will come with a lot of baggage in terms of doing epsilon balls and rejection sampling. And typically you have at the end a system that doesn't scale to high dimension like the system we show here. And so that's why we pick F carefully so that we can approach this modified target using a Markov chain. And so at a high level, so this Sitka work is using a perfect phylogeny plus an error model. And what is maybe the most interesting is that there's this new MCMC move that's specialized to this model. Just to give a high level overview of what it allows us to do is if you compare it with classical phylogenetic, Bayesian phylogenetic ways of exploring the tree space, like nearest neighbor interchange or the subtree prune regraph, this method that we developed has a lower computational cost. So it's the cost grows like the sum of the number of cell and number of loci, whereas these other methods, they scale as the product. And even more striking is that the size of neighborhood, the number of trees around the current tree that we can score quickly, is exponential, whereas it's 3 for the nearest neighborhood near NNI move and L for the SPR move. So this transformation brings in an approximation, but then allows us to scale computation. So to give you a bit of an example of what is output from this, so we get a consensus tree that summarizes a posterior distribution over trees. And what we're showing here is a certain representation of uncertainty. So you can, each of those markers or loci that I called the previous slide on the different chromosome, there are locations where there was a critical mass of change points, so that there's an interesting posterior to show. So they come in triple. So in black is whether the data we observe jump or not in copy number. And then what I show in red is the posterior. So how confident the model that there is actually a change here in terms of having a clade in the tree that has a mutation at that contains that loci here. And finally, there's the consensus reconstruction in orange. So by comparing what the actual is, the actual data to the reconstruction, we can get some kind of goodness of fit from this output. And the posterior can also give us a notion of calibration that when it's very confident, we would expect to see that the data is actually present there. And so, of course, we started by looking at synthetic data. In phylogenetic tree, you don't observe the true tree, so that's the big challenge of this problem. So you have to start with synthetic data, and on synthetic data, our method works quite well. But we wanted to go further and look at how well it performs on real data, something that's really challenging in general in phylogenetics. But because of this goodness of fit strategy that I briefly described in the last slide, we can provide some quantification of the goodness of fit of different methods. And again, the method had an edge over competing method on 2 triple negative breast cancer data set and 1 high-grade serous ovarian primary. We looked a little bit also, since we know that there are some violation of these infinite site assumption, we can simulate data that have these violation at rates that we estimate based on heuristic on how much of these violations we think there are in real data. And the model degrades gracefully thanks to the error model. Yeah, so the model yes. So I'll start just mention quickly then some results. You can then use this inferred tree based on only copy number data to ask the question, now, if we reintroduce the single nucleotide variants, can we place them on the tree? So for some of them, the answer is no, because if we have a SNV that arose close to the leaves of that tree, because of the way the single cell sequencing works, there will be very, very few reads, so we can't really place it. But the idea is that if it occurs high enough in the tree, it will actually present in a big enough number of cells, some sort of pseudo bulk subset that allows us to place that mutation. So what we did here, so we fixed a tree inferred by from copy number, and then we asked them all, okay, so place each of those SNVs on the tree, and then we can see what is the actual data. So that's the raw data, and we can see that the SNV data recapitulates these large clades, and they seem to suggest some sort of serially branching trees within the clade. However, it's hard to tell exactly what's going on, given the small number of cells that differentiate these lower part of the tree. So maybe I should end here. So, or I maybe how am I doing with time? Maybe is my question. I can stop here easy.
So there's about 5 or so minutes left. We usually end on the hour in case anyone has other classes to go to.
Perfect. So I'll use 4 minutes to go briefly about some ideas of what's going on under the hood. So how do we do Bayesian computational approximation of these complex posterior distribution? So the high level problem here is that we have often challenging posterior distribution. So the toy example you often see is like, what if you had a bimodal distribution and you can only move in a neighborhood, then you sort of get stuck in one of those modes. So as the number of MCMC iteration goes, you stay in one mode and you miss the other one. So these problems, they do occur in real data. So this is another model that I haven't had time to describe, but this is for doing even more closer to the data. So the first step, which is to infer the copy number. And so we can see here the raw data, so the log GC contents and the log read counts. And you can see this is a signature of different copy numbers that's occurring here. Now, what's interesting is that in some of these cells, there are some weak evidence that there might have been a whole genome doubling recently. And so what you see is for most of the genome copy number, the copy number can be explained with the basic assignment of, this one being 2, this one being 3, and so on. But there's a few regions where there seems to be a count that would like to go at, between 2 integers. So there's just a few of those that seem to suggest you should actually double the whole genome, but it's very noisy. So there's sort of this bimodality of, is it that there was a recent doubling, or is it just noise that's creating these in-betweens? And so to approach this problem, we look at recent tools that instead of just using one distribution, use a continuum of distribution. So the standard example, but we're going beyond that as well, is annealing. So you can imagine at one hand, you sort of have the likelihood of the tree that's toned down all the way to 0. So you just have the prior distribution. And the other extreme is the posterior. So often the prior is something we can sample from IID. We would like to basically have communication between these two to propagate, to be able to switch mode, for example, in posterior sampling. So how do we go from a continuum of distribution to an inference algorithm? Well, we discretize it. So we look at, instead of a finite number of chains that approximate this, discretize this continuum of distribution. And so there are MCMC Move for each of these chains, but crucially, there are also some moves that swap, that propagate information between. So what we found recently is that if we use non-reversible methods to do that propagation between different distribution, we can get much higher, provably higher performance than previous algorithms. And what we see then is really this kind of behavior where by going to this easy distribution, we can jump to this other mode, both in toy data, but importantly in real data. Now, the key thing why we need to talk about the continuum is because the discretization is very critical. So you need to find an adaptive discretization that allows us to interpolate between these two, the easy distribution and the hard distribution. So that's some of our recent work is based on better discretization for these continuum of distribution. So I'm not that. So just maybe like the 30-second version is that what we observe is that if you look at the difficulty of the problem, so this is a continuum of distribution from prior to posterior. Often what we see is there's a phase transition, a certain point in the middle where the problem gets in the neighborhood very difficult, and we need to allocate more discretization point here. So these kind of phase transitions, they're well known in physics, but what we found is that they're actually very present in Bayesian inference problem, for example, in phylogenetics and inferring ODs and all different problems. And so another thing that we're looking at also is taking shortcuts. So instead of using just the geometric annealing between the two distributions, we're building other continuum of distribution. So for example, adding one leaf at the time in a sequence of trees and annealing just that one leaf that we just added, or using more generalized notion of annealing that I'm not gonna have time to go over. So just a kind of a taster of the kind of work we do at the computational front. And basically this translates to real improvement in biological problems. So going back to this problem of inferring copy number, we see that older discretization method, they had a lot of problem switching between these two explanations. So either not doing a genome doubling or not. MCMC is hopeless. And our advanced adaptive method can efficiently switch between these two explanations. So thanks everybody, and I'm happy to answer questions.
Thanks very much indeed, Alex. I think in the interest of time, Lorenza, could you confirm that we have the same website, same link for the next hour?
Yes, we do.
So perhaps we could use the first few minutes for any questions that anyone has. People who need to go, please feel free to go. And if it's all right with you, Alex, we can start the questions then. Before we do that, I just want to make one announcement, and that is that the next talk in this series is on February the 17th at 4 o'clock, usual time. The speaker is Susan Holmes from Stanford, and she's talking about statistics and geometry for heterogeneous data. So with that, I'll open the floor to any questions, and we'll use a few minutes, and then the people who are going to meet you in more detail can do that afterwards. Thanks, everyone. Any questions?
Hi, I'm Alexandre Bouchard-Côté from the National Cancer Institute. I wanted to ask you a question about the comments you made about programming languages at the beginning. So I understood that for modeling purposes, you want programming languages that are more expressive and adapted to the problem domain. And then when you're doing your MCMC or your intensive sampling, you want programming languages that have a fast implementation. So are you doing some sort of mixed implementation using multiple programming languages for different parts of your software?
Yeah, so I talk a little bit about this. So, and in some sense, I almost wish that probabilistic programming language were not called programming language. They're sort of maybe the only sort of well-known programming language that is sort of in the same flavor is SQL. So it's the goal of these languages to be at a very, very high level, so high that the programs would actually look like mathematical statement of this form. And under the hood, these languages have, well, they have right now they have trade-off. Like often you can get as much computational performance as handwritten code, but as time goes, they become more and more viable for large-scale analysis. This specific one that was developed is hosted in the JVM. So there's sort of just-in-time compilation that goes down the pipe. And a lot of the, so every language, programming language now is, and I should mention another one that's quite popular is the WinBugs language. So I guess that's kind of the ancestor of all this stuff, at least in the statistical world. It's trying to put these bugs language to the next step, where like you can have not only integer and real, but also trees and network and that kind of stuff. So as a user, at least the time it takes to write programs is short. But the trade-off is that it's not as fast as handwritten code. Yes. Does that answer the question?
It articulates the trade-off. I'm asking what you're doing for the software you described. Are you sort of hand coding the time-intensive parts and using these programming languages for the not time-intensive parts, or?
Yeah. So for the language I would develop, Blang, so there's a category of POSIX programming language that attempts to use a universal inference engine that is always the same thing that's used. And so you don't have to write any inference code, but it's really slow. So Blang is trying to do a trade-off, which is essentially each data type has to have its handwritten inference algorithms. But then if we want to try several priors or several hierarchical model, once there's a sampler for a given type, then it's easier to write code.
Thank you.
Yeah.
Any more?
So a quick question. So thank you for your great talk. And I have a question. So at the beginning, you say your goal is to characterize the fitness characteristics of subpopulations inside the tumor. But I was wondering, why not just say, study the cell type distribution or such kind of distributions, the dynamics of such kind of distributions to directly understand evolutionary processes within a tumor? Well,
the advantage of going to this Wright-Fischer framework, I think, is really that we can, it's a foundation to build more sophisticated models. So first of all, we get something that we, it's grounded in a large literature. So in population genetics. So it bridged this important literature. And then it creates a foundation where if, let's say, we want to add a bottleneck effect. So we know that there's a bottleneck each time we do an engraftment, then we don't have to start from scratch. We can look at the population genetics literature and what are the best, the models that are used to extend the Wright-Fischer to take into account bottlenecks. So that's, I think it's advantageous to look at the population genetics literature and see what they do and build from that. But yeah, so I'm not sure if that answered the question, but that was the motivation, I guess, for using the Wright-Fischer.
Mm-hmm. Any other questions?
I had a quick question. Hi, Daniel Temko. Thanks for a great talk. I was wondering, did you think about trying to infer the fitness of mutations rather than groups of cells?
So that's something that there's a fundamental limit of what we're going to be able to accomplish here, which is that all the mutations that occur within one edge of the tree that we can resolve, we cannot go deeper than that from this framework. So all we can say at this point is basically we can go. We're not able yet to go at the point of one edge, but basically a sub part of the tree where we say there seems to be something that happened there, and but beyond that, we need to look at other things like intervention, CRISPR-Cas9 intervention, or some other platform to be able to disentangle which of those, even if it's copy number driven or if it's point mutation driven or something else.
Thank you very much. Makes sense.
Okay, so I think what we should do is revert back to the original plan and let the people who are going to have a more in detail chat to Alex get on with that. But thank you very much, Alex. That was wonderful.
Thank you. Thank you so much.
Our pleasure.